Hello, everyone, and welcome to the Basic Sciences Oral Abstract Session number 1. Bonjour et bon après-midi tout le monde et bienvenue à la séance en sciences fondamentales, la séance de présentation des orales d'abrégé numéro 1. Our first presenter today of a really outstanding lineup is Julien Klein from Université de Laval. Julien, whenever you're ready, please go ahead.
Thank you. Do you see my screen?
Yes, you're good to go, Julien.
Okay, it's perfect. So good afternoon, everyone. First of all, I would like to thank the CAHR members for allowing us to attend the conference despite the pandemic and for giving me the opportunity to present. Today, we are in the area of antiretroviral treatments, identification of viral reservoirs, and improvement of therapies. So in this presentation, I will talk about the impact of early antiretroviral therapy on tissue-resident myeloid cells in the liver and lung of HIV-infected rhesus macaques that are used as a model in our study. I don't have any conflict of interest to disclose in this presentation. For a fresh reminder, one of the major problems in the HIV field is the misunderstanding of viral reservoirs under antiretroviral therapy. For instance, in our last study, we showed that the administration of early treatment drastically reduces the virus in the blood to an undetectable level, but when the therapy is interrupted, we can systematically observe a viral rebound that indicates the presence of viral reservoir in deep tissues. And our data confirm the study of Whitney et al., who treated individuals at day 3 post-infection. As we know, HIV and SIV can target lymphoid cells like CD4 T cells and myeloid cells that are heterogeneous populations. In our last study, we focused on the contribution of short-lived circulating monocytes in viral reservoir establishment. Circulating monocytes are from bone marrow. They can be infected by HIV or SIV, and they are particularly present in the blood, the spleen, and the intestines. In the lab, we showed that in early ART-treated monkeys, short-lived monocytic cells do not contain viral DNA, as shown here, suggesting that they do not represent viral reservoir after early ART. However, little is known so far about long-lived tissue-resident macrophages, called here TRMs. Studies in mice have shown that the liver and lung also contain tissue-resident macrophages, which are not derived from bone marrow, but from the yolk sac. And to characterize these cells, several membrane biomarkers are proposed, such as CD44, TIM4, CD117, CD206, CD64, LIV1, and CD200R. That's why my research now focuses on the role of tissue-resident macrophages during SIV infection. So in order to achieve this, my 3 objectives are to determine the viral infection in the liver and lung of our model of HIV-infected rhesus macaques, and then to characterize tissue-resident macrophages in the liver and lung, and finally, to evaluate the impact of early ART on liver and lung infection. So how do we proceed? We used rhesus macaques that we naturally infected by HIV and macaques that we treated at day 4 after infection. Monkeys were sacrificed at different time points post-infection, and we recovered liver and lung tissues for viral quantification, for viral RNA visualization, for tissue-resident macrophages characterization, and for transcriptomic analysis. And for comparison, we performed the same experiments with splenic tissue. So in objective 1, when assessing viral infection in the liver and lung, our results demonstrated that liver contained comparable levels of viral DNA and RNA, while in the lung, here, we detected significantly higher levels of viral RNA than DNA. Interestingly, these HIV RNA levels in the liver and lung are both strongly positively correlated with the viremia of infected monkeys. Moreover, in situ hybridization showed that the spots of viral replication, as shown here and here, are located in perivascular zone where circulating cells infiltrate the tissue. We next performed deep mRNA sequencing of liver and lung tissues of 2 infected monkeys compared to 2 naive monkeys. And our results demonstrated that both infected tissues show a highly inflammatory profile with an increase of inflammatory response markers, an increase of myeloid cell markers, as well as interferon-stimulated genes markers consistent with viral replication. Next, we assessed the characterization of tissue-resident macrophages. We then performed cell surface stainings, and we analyzed HLA-DR and CD11b positive cells. And the phenotypes of tissue-resident macrophages in infected monkeys indicated that CD117, CD206, and LIV1 could be markers of interest to define specifically tissue-resident macrophages because they are highly expressed in the liver and lung, which is not the case in the spleen, as shown here. Interestingly, the same cells, the same tissue-resident macrophages co-express the CXCL10 receptor, CXCR3 or CD183. And this receptor is involved in cell recruitment to inflammation sites. And interestingly, the cells also co-express CXC3 CR1, the fractalkine receptor that is not shown here, but this data suggests that tissue-resident macrophages also exhibit an inflammatory profile. Now, to assess the contribution of tissue-resident macrophages during HIV infection, we performed cell sorting by flow cytometry based on CD206 marker specific for tissue-resident macrophages and CD14, a circulating myeloid marker. And our results showed that individuals in whom viral DNA is detected in blood monocytes, CD206, but also CD14 non-CD206 subsets, both contain viral DNA in the liver and the lung. On the other hand, subsets that do not contain viral DNA, here and here, correspond to individuals in whom no viral DNA was detected in blood monocytes. So this data suggests a heterogeneity of the infected populations in these tissues. Next, having observed viral infection in the liver and lung, we assessed the impact of early ART on liver and lung infection. The monkeys were infected and treated at day 4 post-infection with a cocktail of antiretroviral drugs during 8 weeks, sorry. 5 monkeys were sacrificed during this period, and at day of euthanasia, no viremia is detected in all of them, as shown here. And first, transcriptomic analysis showed that early ART significantly decreased, as shown here and here, transcript of pro-inflammatory cytokines, myeloid cells markers, and interferon markers, demonstrating that early ART prevents inflammation and the recruitment of myeloid cells. We then quantified viral DNA and RNA in the liver and lung in parallel with circulating monocytes. And our data showed that no viral DNA and RNA is detected in circulating monocytes, as well as in total liver and lung. Thus, we highlighted that concomitant with the absence of infection of circulating monocytes, early ART leads to the absence of viral infection of the liver and lung. In conclusion, we propose that in our model, HIV infection of liver and lung is associated with an important inflammation and a recruitment of infected monocytes. Once recruited to inflammatory sites, infected monocytes differentiate into inflammatory macrophages, acquiring tissue-resident macrophages markers. Thus, viral DNA found in tissue-resident macrophages are comparable to those found in monocytes. Finally, given that we provide evidences that early ART prevents early viral seeding both in the liver and lung, concomitantly by preventing inflammation, we highlight the crucial importance of early treatment by decreasing anatomical viral reservoirs. To conclude, I would like to thank my director, Jérôme Estaquier, the lab members, the funding members, but also the monkeys without whom this work would have not been possible. And thank you for your attention.
Thank you very much, Julien, for that excellent and very clear presentation. You can use, for the attendees, you can use the Q&R function on your browser to ask us questions. But I see the other Julien perhaps has a question first. Maybe Julien, would you like to go ahead? Van Gravenage?
Do you hear me?
Yes, go ahead.
So really great presentation, Julien. I was wondering whether or not there is some evidence about the impact of early treatment on the reservoir in the major reservoir, aka the lymphocyte, the memory T cells, the CD4 T cells.
Yes, of course. The last year, no, in 2020, we published the papers about the spleen and mesenteric lymph nodes, and we found that despite early ART, we found viral DNA in TFH cells and effector memory cells of mesenteric lymph node and the spleen. And it was not the case in peripheral lymph node. So the major finding here is that early ART do not prevent the viral seeding in the spleen, in TFH cells and effector memory cells in the spleen and mesenteric lymph nodes. And the last year, in December, we published the articles about the brain, and we found that with Chris Power, Dr. Chris Power, we found that early ART do not prevent viral seeding in the brain too, and more particularly in the microglia and macrophages.
Okay, thank you very much.
Thank you, Julien. Dr. Nicole Bernard, would you like to ask your question? Go ahead, Nicole.
Yeah, from what I understand, at the very beginning of your talk, you were saying that if you started ART 4 days after infection, it didn't prevent the infection of CD4 T cells. So what accounts for the later window that you have to infect for monocytes, such that, anyways, it sounds as though you can wait 4 days to start ART, and that can prevent the establishment of infection, whereas really the window is only 3 days if it's CD4 positive T cells, if I understood correctly.
At the beginning of my presentation?
Yes, you talked about your previous work with CD4 T cells?
Yeah, with CD4 T cells, yes.
So you showed that if you start ART at 4 days,
yes.
It doesn't prevent the establishment of infection, but the work from Whitney,
yes.
they had to treat by 3 days in order to prevent that.
They treated at 3 days. They treated at 3 days, and it did not prevent the viral seeding too. because there are viral rebound in any cases in monkeys.
Okay, so it's the same thing. 3 days,
Yes.
it prevents infection, 4 days.
Study. Yes, 3 days and 4 days at the end, it does not prevent viral seeding, viral reservoirs.
Okay.
So it's too late. Yes.
Okay. Let me read one final question from the audience. First of all, they say, great presentation, Julien. Did you observe a clonal expansion of infected tissue-resident macrophages in these macaques?
It's in process.
It's in process. All right, we'll be waiting. Like by the end of the session, you think? No.
No.
Right? All right.
Because I don't have the results now, but it's the goal. Finally, it's the goal. You know.
Okay. Thank you. Let's all thank Julien for an outstanding talk. Let's give him a round of applause. Thank you very much.
Thank you so much.
All right. Our next speaker is Alexis Hérault from the Université du Québec à Montréal, UQAM. Alexis, the floor is now yours.
Hello, can you see me?
Looks good, Alexis. Yes.
Okay, so hello everyone. Thank you again for the invitation. And today I will be talking about the dynamic of regulatory CD8 T cells in acute HIV infection and following early ART initiation. We have nothing to disclose. We have no conflict of interest in this project. So FOXP3 expressing immunosuppressive CD8 T cells are less studied than their counterparts CD4 Tregs, mainly because in physiological condition, the expression of FOXP3 by CD4 T cells is higher than CD8 T cells, and also the frequency of CD4 Tregs is higher or more elevated than comparing to CD8 Tregs. This, along with the lack of well-defined cell markers to identify CD8 Tregs, have made that the scientific community have not that big interest in the study these cells. However, CD8 Tregs have an undeniable role in cancer, autoimmune diseases, transplantation, and infectious diseases. In this case, CD8 Treg frequencies increase during untreated HIV and SRB infection, and this increase could contribute to disease progression by inhibiting the anti-HIV specific responses, promoting tissue fibrosis via TFeta production, and also contributing to viral persistence. Nowadays, it is recommended to start treatment very early, since early AR initiation promotes CD4 T cell recovery, immune reconstitution, and also decrease the size of the viral reservoir. In this sense, we recently reported that in a model of SIB infection in rhesus macaque, that untreated SIB infection promote an increase in the frequency of total CD8 Tregs and CD39 positive CD8 Tregs that were normalized after 4 days by early AR started at 4 days post-infection. But the comprehensive characterization of CD8 Treg subset during the acute HIV infection and following early AR initiation is still needed to assess. So to evaluate the dynamic of CD8 Tregs and the effect of very early AR initiation during HIV infection, we use samples from cohorts of people infected with HIV here in Montreal, particularly the Montreal primary HIV infection cohort and the Canadian cohort of HIV infected slow progressor. In our cross-sectional study, we included a total of 85 individuals that were divided into 5 groups, including uninfected, as a control, acute and chronic infected individuals, are treated, and elite controls. We also assessed 25 individuals prospectively, and 10 of these individuals were treated in the acute infection, and the rest remained untreated. Thus, the effect of earlier initiation was assessed in the longitudinal study. First, we look at the frequency of total CD8 Tregs and we identify these cells by the expression of FOXP3. Here, we observed that untreated SIB infection was associated with an increase of the frequency of CD8 Tregs, and there was no difference between elite controller and uninfected control. Earlier initiation normalized the frequency of these cells. Then we look at the effect of immune activation and immunosenescence, or the effect of the infection in CD8 Tregs immune activation and senescence. And in the case for immune activation, we use the expression of a well-known marker of activation, HLA-DR and CD38. And for immunosenescence, the phenotype CD28 negative, CD57 positive was used to identify senescent CD8 Tregs. Here we observe an increase in the frequency of activated and senescent CD8 Tregs. And only in the case of senescent CD8 Tregs, we observe an increase in elite controllers comparing to non-infected individuals. In earlier treatment was unable to restore the burst frequency, so was unable to normalize the frequency of immune activation and senescent CD8 Tregs. But what happened with the immunosuppressive subsets? So we know that there are different immunosuppressive subsets. And first, we look at the expression of CTLA-4 and PD-1 that are immunocheckpoints that are both well known for being needed for the immunosuppressive function of CD8 Tregs, and both also contribute to CD8 Tregs expansion and proliferation by inducing FOXP3 expression. Here, we observe an increase during untreated HIV infection in both CTLA-4 and PD-1 CD8 Tregs, and no difference were observed in comparing elite controllers and non-infected individuals. Earlier treatment only restored the frequency of CTLA-4 positive CD8 Tregs. Then we look at the expression of two other well-known markers of immunosuppression, in this case CD39 and lab TGF-beta1. CD39 is an ectonucleotidase that, together with CD73, that is another ectonucleotidase, converts the inflammatory ATP into anti-inflammatory and immunosuppressive adenosine. And also the expression of CD39 by Tregs have been linked to disease progression. On the other hand, TGF-beta is also well known to be needed for the immunosuppressive function of CD4 and CD8 Tregs. And importantly, TGF-beta stimulate the production of collagen-1. The collagen-1 will deposit and will promote the tissue fibrosis. And of note, there is a functional interplay between CD39 and TGF-beta in the context of fibrosis. Here, we observe an increase in CD39 positive CD8 Tregs and lab TGF-beta positive CD8 Tregs in untreated HIV infection, while no difference were observed for both populations in comparing elite controllers and non-infected controls. Earlier treatment initiation only restored the frequency of lab positive TGF-beta Tregs. As you know, the gut-associated lymphoid tissue, or GAL, is the main site of HIV and SIV replication, CD46 depression, and lymphoid tissue fibrosis, which start very early during the acute infection. So we wanted to evaluate the potential migration of CD8 Tregs toward the gut. For that, we evaluate the expression of two well-known markers of common migration to the gut, in this case, interleukin beta-7 and CCR9. We observed that an increase in both interleukin beta-7 and CCR9 positive CD8 Tregs during untreated HIV infection. So that means that the potential migration towards the gut of CD8 Tregs is increased during untreated HIV infection, and no difference were observed between elite controllers and non-infected individuals for both markers. And earlier treatment initiation failed to reduce or to normalize the frequency of both sources. Finally, we were interested in evaluate what happened with the CD39 and lab TGF-beta positive CD8 Tregs that could potentially migrate to the gut. So for that, we evaluate the expression of CCR9 and interleukin beta-7 in CD39 positive and lab TGF-beta positive CD8 Tregs. We found that in all of the cases, the untreated HIV infection was associated with an increase in the potential migratory capacity of these immunosuppressors and highly fibrotic CD8 Tregs, and early R initiation only restored the frequencies of interleukin beta-7 lab TGF-beta positive CD8 Tregs, while was unable to normalize the rest of the population. We can conclude, or we showed that early R initiation did not normalize the frequency of immunosuppressors and pro-fibrogenic CD8 Tregs, and their potential migration to the gut, which could contribute to immune dysfunction, gut fibrosis, and HIV disease progression. On the other hand, elite controllers shares most of the CD8 Treg characteristics in infected individuals, which could be related to the maintenance of effector cell functions and further better viral control in this individual compared to HIV progressors. I would like to thank to all the study participants, to our collaborators in the research room, Marquil and Resocida, and also the funding institution of both my scholarship and the project. And thank you all of you for letting me be here and present.
Thank you very much, Alexis, for that great talk. If there are any questions at this time, we have a few minutes to ask Alexis questions. Okay, I have lost my screen on the left. Just a second here. Oh, Julien, please go ahead.
Yeah, for sure. Really great presentation, Alexis, and really great data too. I was wondering, because you find the differences between the different groups of PLW of 0.5% to 1%. So since you are not looking at HIV-1 clones, I was wondering what is meant biologically speaking, because in the papers that we are referred to, when you say that those Tregs, CD8 Tregs are immunosuppressive, what is the proportion in this paper of this population?
Yeah, so if CD4 Tregs compared to CD8 Tregs are in physiological condition, and most of the report like 40 to 50-fold high than CD8 Tregs. So normally the CD4 Tregs are between 5 to 10%, could be higher during some condition like cancer of in HIV, for example. And CD8 Tregs normally in physiological condition is up to 1.5%. And here we observe, you see in healthy around what is expected, less than 1%, and during the infection are increasing in the presentation of these cells.
Okay.
And when we look at the subpopulation, as we are seeing in the total CD8 Tregs, in the pool of total CD8 cells not inside FOXP3 expressing cells, of course, you dilute the population because you are seeing one population that per se is small, the CD8 Tregs, and then you are combining more markers. So when you see the total percentage, we see total CD8, that's why you see a small percentage.
Okay, great. Thank you very much.
Thank you very much. Just a reminder to use the Q&A box on the platform if you have questions that you want to ask the presenter or anyone else. Okay, maybe one short question for me, Alexis. Can you, maybe I missed this, but you could just perhaps identify how you characterize the elite controllers in the cohort? How were they characterized primarily?
Okay, so elite controllers are individuals that without treatment, they control the viral load, have undetectable viral load, and maintain the CD4 cell count. I can show you. I mean, I have this table, but I didn't present it. So these are the characteristics of the, let me, wait. So these are the elite controller groups. We have 18 individuals. They were really old comparing to the other groups, and most this is this they maintain. You see, comparing to they have the similar. There is no difference with the CD4 cell count about the with comparing to non-infected. CD8 cells was higher in this case, and there was no difference as they were different in the CD4-CD8 ratio. But that could happen. That could be related with the age, with the sex. There are different impacts on this, and they have higher nadir than the other HIV infected groups.
Okay, thank you very much. There are no further questions. Let's thank Alexis for an outstanding presentation. Thank you very much. A round of applause. All right. Our third presenter this afternoon is Tram Pham from the Institut de Recherche Clinique de Montréal, the IRCM. Tram, please go ahead and share your screen.
Thank you. Thank you very much. Good afternoon, good morning, everyone. We thank the organizers for the opportunity to present our work at this meeting. So over the past many years, different humanized mouse models have been developed for different lines of research. Perhaps one of the simplest models has been the humanized energy, whereby human stem cells are injected into immunodeficient mice of the NSG background. And in this system, there is partial reconstitution and function of human cells, but there's really no maturation and education of T cells because of the lack of a thymus, of the human thymus. So to address this issue, The humanized BLT, or bone marrow liver thymus model, was developed, whereby pieces of fetal thymus and liver are implanted under the kidney capsule of the NSG mice. Human stem cells are also injected in these mice. And in this situation, there is significantly more robust reconstitution of human cells, and there is superior development of functional immune cells. And for the past many years, it has been considered a gold standard small animal model for HIV research. That being said, there are practical and ethical challenges associated with the use of fetal tissue. And in this context, it has been shown recently that instead of using fetal thymus fragments from neonates that is taken from cardiac surgery can be used for the engraftment of mice, as shown here on this line. An obvious advantage of this approach is that the size of a neonatal thymus is significantly larger, making it possible to make up to 50-fold more BLT mice compared to the classical approach. So with this, the goal of this study was to determine if BLT mice made with cardiac surgery thymus can support functional T-cell development and be a valid model to study HIV infection and persistence. And for the purpose of this talk, I will refer to BLT mice that are made with cardiac surgery thymus as CCST and BLT mice that are made with fetal thymus as BLT. So before diving into the results, I would like to mention that in our study, the engraftment site of cardiac surgery thymus is the quadriceps muscle, because our previous attempt at implanting the tissue under the kidney capsule, as was done in the new thigh model, proved repeatedly unsuccessful. I would also like to say that we have been able to use biobank cardiac surgery thymic tissue from up to 5-year-old donor, as opposed to the 10-day-old donors, as in the case of new thigh model. When we look at the level of human cell reconstitution in these mice over a period of 30 weeks post-humanization, we find that the reconstitution of total human immune cells, which is CD45 positive, is significantly higher in CCST mice and BLT mice compared to the NSG mice. We see less T cell in CCST mice compared to BLT mice, and the converse is true for B cells. However, in order to be sure that the T cell in CCST mice come from the engrafted CD34 positive human stem cells, we transduce these stem cells with a GFP expressing lentiviral vector, and 24 hours later, we administer these cells to the mice. And as you can see from here, at 7 weeks post-humanization, there is a transient mix of T cells that are coming from the CD34 positive HSC, but also T cells that come from the implanted thymus. However, after this period, basically starting at 8 weeks and beyond, the T cells that are present in CCST mice largely come from the CD34 positive human stem cells. The fact that we have a similar frequency of GFP positive cells in the fraction of T cell, in the population of B cells, and in the population of CD14, starting at 9 weeks post-humanization, really indicate that the T cells that are present in CCST mice do really come from the engrafted CD34 positive cells. We then wanted to look at the functionality of these T cells in the CCST mice. So we perform an allogeneic tumor challenge experiments by looking at the ability to reject tumor cells which express luciferase. And we perform the analysis using bioluminescence. And as you can see from here, NSG mice are unable to reject tumor cells and die within 7 weeks of injection. BLT mice, on the other hand, are able to completely reject tumor cells and survive over the entire period of the experiment. The response in CCST mice was a bit variable in a sense that about 40% of the mice were able to completely reject tumor cells and survived, but the other ones did succumb to leukemia, although at a slower rate than the NSG mice, as shown here on the blue line. We also wanted to look whether to see whether these T cells were able to make interferon upon mitogen stimulation. And indeed, as you can see from here, after PMA alnomycin stimulation, the T cells in CCST mice were just as capable of making interferon gamma as the T cells in BOT mice. We then went on to see whether the CCST mice are susceptible to HIV infection. So for this, we infected these mice with a CCR5 tropic HIV, and then we followed them for a period of 7 to 8 weeks. So as you can see from here, at the level of viremia, CCST mice, as shown here in the blue line, are just as susceptible to HIV infection as BOT mice. And this is independent of the route of viral inoculation. At the level of virus expression in the tissue, we find that the frequency of P24 positive T cells in CCST mice is as comparable, if not higher, than that observed in BOT mice. We then wanted to see whether T cell in HIV-infected CCST mice were able to mount a response in an antigen recall assay. So indeed, when we activate these T cells, both CD8 and CD4 T cells, with a pool of HIV peptide, we see that T cell in CCST mice were more capable of making interferon gamma compared to the T cells in BOT mice. We then went on to see whether this model could be used to characterize the development of HIV reservoir. So to do this, we infected the mice with the HIV as we showed previously. At 6 weeks post-infection, we treated them with combination antiretroviral therapy. And as you can see, in response to our treatment, viral suppression was observed in both models. We also see a complete reduction of P24 positive T cells in the tissue of mice that were treated with antiretroviral therapy. And importantly, we also see a significant decrease in the frequency of cells that harbor the integrated HIV DNA genome. So lastly, we wanted to see whether CCST mice could be used as a model to study HIV reservoir. So to do this, we infected the mice with HIV as before. We treated them with ART for 6 weeks as before. And then after that, we removed the medication and we let it go for another 6 weeks. So as you can see from here, upon treatment interruption, the virus rebounded to the level of even higher than before treatment in most of these animals, suggesting the presence of HIV reservoir in this system. So lastly, I would like to conclude the talk by saying that CCST mice support functional T cell development. They are highly susceptible to HIV infection and enable studies of HIV persistence during ART. The sheer quantity of a cardiac surgery thymus allows for a larger production of CCST mice compared to the conventional BOT mice. And with that, we believe that CCST mice represent a practical and ethical alternative to traditional BOT mice made with fetal thymus, not only for HIV research, but also for other applications. And with that, I would like to acknowledge some members of the lab who assisted us in this study. We'd like to thank Gilead and Merck for providing us with the compounds used in this study. We thank, we thank the St. Justine Cord Blood Biobank. We would like to thank Dr. Jonathan Angel and his staff in Ottawa for providing some of the precious material used in this study. we like to thank the staff of the animal care facilities, both at the ISCM and St. Justine, for taking care of our mice. Of course, we would like to thank the funding agencies who provided the funding for this work. And I thank you for your attention.
Thank you very much, Tram, for that great talk. We will now take questions. Nicole, do you have a question?
Yeah, I do.
Go ahead, Nicole.
Very nice talk, very nice piece of work. I'm curious about the survival of the mice, the BLT versus these cardiac surgery engrafted mice. Is there a difference in how long they live? And I know that the BLT mice don't differentiate NK cells, so
no.
Do the cardiac surgery mice do?
No, they don't. They don't. I think that we have to have things like IL-15 to facilitate the development of NK cells. In terms of the survival, I think that both of them, like over the course of the experiment, of course, you do lose mice, but I would not say that the CCST mice are more prone to death than the BLT mice.
Thank you.
Okay, we have some questions from the audience. First question, did you observe HIV DNA harbored in cervical mucosa of infected BLT mice?
Okay, so I have not looked at that in this particular study, but I think that someone in the lab previously was trying to look at that, but I don't think that the results were really conclusive. So I wouldn't be able to say yes or no at this point.
All right, thank you. We have a second question from the audience. What is your source of hematopoietic stem cells? Do they match the thymus HLA?
No, they don't. And we find that like in our study, like the HLA compatibility doesn't really have a big impact on the ability of on the success of generating the CCST mice.
Thank you. Okay, I think Julien had a question from our room. Julien, do you still have a question?
Yeah, for sure.
Go ahead.
Really great presentation, Tram. I was wondering, how can you explain the discrepancies between half of your population CSST regarding the reject of the tumors? Because it's the same procedures, it's the same strain, the same tissues, basically. How can you explain that some of these CSST mice reject tumors in your model of leukemia versus the other one don't?
So for us, this is what we find with the CCST mice. We don't know exactly the reason for that. What we notice is that with the CCST mice, the variation between mice tend to be a little bit more than with the BLT mice. And that's why, like within the same batch, but we have some that are able to completely reject, but some they don't. Like basically, they are less they're less able to
and
to
those the mice which doesn't reject, but which don't reject the tumor, can be considered as a good HIV infection model still? Because it's CD8 related, the graft reject, no?
Yeah, so for us, we don't see all of them, like, they are still able to be susceptible to HIV infection. So right now we're not able to, there's no evidence to say that, okay, for the ones that are not able to reject tumor, they're not susceptible.
Okay, thank you very much.
Okay, let's all thank Tram for an outstanding talk. Let's give her a round of applause. Thank you.
Thank you.
All right, our next presenter is Sankit Kant from the McGill University Health Center. Sankit, whenever you're ready, the floor is yours.
Okay, thank you. I hope everybody can see my screen. No
Looks
black
good,
bars.
Sankit. Yep, everything is great.
Excellent. So, hello everyone. My name is Sankit. I'm a PhD candidate in Nicole Bernard's lab at McGill University. And I would like to thank the organizers at CAHR and the selection committee to select our abstract for this presentation. And today I will be talking about antibody-dependent functions and the biophysical characteristics of non-neutralizing antibodies in elite controllers that are associated with spontaneous control of HIV. We don't have any conflicts of interest to declare. This project stems from the only successful protective vaccine trial that took place in Thailand, the RV144 trial, where antibody-dependent cellular cytotoxicity, ADCC in short, was one of the correlates of HIV protection. But there are other antibody-dependent functions similar to ADCC, and whether they play any role in HIV control is currently unknown. So for understanding or investigating these mechanisms for spontaneous HIV control, for our study, we recruited 4 groups of people living with HIV. They are untreated progressors who are not undergoing any treatment, as a consequence have very high viral loads. Treated progressors are individuals who are going under a successful therapy, which as a consequence, they have viral loads below the levels of detection. Elite and viremic controllers are individuals who can spontaneously control their viremia without any treatment. However, elite controllers have very low viral loads, which are usually below the levels of detection. Whereas viremic controllers can maintain stably their viral loads to less than 3,000 copies per ml, as per our study definition. So we collected the plasma from these 4 groups of people with HIV, and we performed these methods as shown over here. So in our lab, we developed sorted HIV-infected CM cells, which are 100% infected, 100% HIV infected, that express envelope in a closed conformation, and they do not express, they downregulate CD4 from the surface of the cells. So using these targets, we measured the amount of antibodies binding to envelope. We also measured ADCC, trogocytosis, and complement deposition, which are antibody dependent. We also measured the phagocytosis function on GP120 coated beads. And because we use these beads, we use GP120 coated wells to measure the GP120 specific antibodies. Thereafter, we collaborated with Dr. Gilly Tolter's lab at Reagan Institute, and we shipped over some samples to them. And they performed complement deposition, phagocytosis, and neutrophil phagocytosis using GP120 coated beads. They also performed 2 biophysical characteristics. The first being glycosylation patterns for GP120 specific antibodies. And they also performed or detected IgG subclass distribution for these 4 groups of people living with HIV for the 5 groups of antigen as shown over here: GP120, GP140, GP70, 41, and P24. So we have attempted with this particular project to perform a plethora of function and to obtain a comprehensive report on the different antibodies characteristics in elite controllers and other people with HIV groups, and to identify specific markers that could distinguish elite controllers from other people with HIV groups. So the first question we had was, is there any differences between the amount of antibodies that are present in these 4 groups? And for us, what we found was that, that the GP120 specific antibodies and the antibodies binding to trimeric envelope were similar in elite controllers, viremic controllers, and untreated progressors, which were higher than those observed in treated progressors. So this was observed in both these assays. And when we measured the amount of functions, we observed similar trends. Elite viremic controllers have similar levels of antibody-dependent functions as untreated progressors, which were higher than treated progressors for phagocytosis, ADCC, and trogocytosis functions. However, we could not observe any specific differences for the complement function. Since there was a trend between the amount of antibodies and the amount of functions in these 3 groups, we normalized the amount of functions with the amount of antibodies per subject on a per subject basis, and we observed that the between group differences had completely disappeared for at least the phagocytosis, ADCC, and ADCT functions, which suggested to us that it's the amount of antibodies that drive the amount of functions. We also measured the reservoir only in controllers, and we could either detect the reservoir or not detect the reservoir. And thereafter, we measured the normalized antibody-dependent functions. And we observed that for ADCC especially, the undetectable group had a significantly higher amount of antibody-dependent cellular cytotoxicity as those from the detectable group. So there was a relationship between ADCC and the reservoir, but we don't know the result and the consequence, who impacts what, is currently unknown from this point of study. One antibody-dependent function could not distinguish elite controllers from others. But when we perform a correlation matrix, what we observed was that elite controllers had a polyfunctional antibody response that was not observed in any other subject group for us. So it was not just one function, but a plethora of functions, an arsenal of functions that are at the disposal for elite controllers that could distinguish elite controllers from other study groups. Then we also measured the antibody, the antigen-specific IgG subclasses between the different people with HIV groups. Each graph indicates the different antigens, red, and each dot indicates a particular subject for the 4 different groups. What we observed was that for IgG2, we observed that unreprogressors had higher amount of IgG2 specific antibodies for GP70, 140, and 41 as compared to elite controllers. And similarly, we also observed that they had higher amount of IgG4 for GP120 and GP41 as compared to elite controllers. Now, we already know from literature from the VAX clinical trials that IgG2 and IgG4 are detrimental or are a marker for inhibiting antibody-dependent functions, and the prevalence of their functions could have led to the failure of VAX clinical trials. So we also wanted to find if there was any relationship between the IgG subclass distribution and the antibody-dependent function. We could not find any noteworthy correlation for untreated, treated progressors or viremic controllers. However, only for elite controllers, the IgG1 subclass was correlated significantly with antibody-dependent functions. But apart from that, IgG3, for example, which is known to be a positive marker, was not correlated with any antibody-dependent function. For the glycosylation, we measured 24 glycoforms. Galit Alder's lab actually did the 24 glycoform classifications, of which 4 stood out. Galactosylation was observed at a higher frequency in elite controllers. These galactosylated antibodies have strong FC receptor or CD16 affinity. Fucosylated antibodies were lower in elite controllers as compared to untreated progressors, and these have a lower FC receptor 3 affinity. So higher prevalence meant that they actually bind low amounts to FC receptor 3. G1F or galactosylated and fucosylated antibodies were prevalent more in untreated progressor. However, the presence of G and the presence of F actually cancel out each other's effects. An interesting observation was that silated antibodies were present highly in elite controllers as compared to treated untreated group. And recently, Dr. Galit Alder's lab published a paper in 2022 itself that individuals with more silated antibodies had a longer time to rebound. So this was a unique marker that we have observed, which is not observed previously published data. Again, there was no noteworthy associations between the markers for glycoforms or the antibody-dependent functions. However, silated GP120 specific antibodies were significantly correlated for elite controllers with the different antibody-dependent functions. So this plays into the previous slide as shown over here, that the silated antibodies have some role or some significance in elite controllers that needs more investigations. So with that, with this presentation and with this project, what our aim was to showcase to the audience was that elite controllers have high amount of polyfunctional antibodies. These amount of antibodies actually dictate the amount of functions, and that there is a relationship between specifically for ADCC and the reservoir. However, we don't know who influences what in this situation or in this story. We also attempted to characterize the biophysical characteristics, but they were not pertinent for elite control in our project. But we need more studies to investigate this objective, in the sense that we need more elite controllers from across the globe who have already obtained these samples to observe if they have observed similar results. With that, I would like to thank the team of Dr. Bernard, especially Dr. Fang Dupi, who was key in developing the SICM cells. Our collaborators, Dr. Jean-Pierre Routy, who developed the primary infection cohort for which we obtained the untreated and treated progressors. Dr. Cecile Tremblay and Dr. Bernard, who enrolled and developed the Canadian cohort of slow progressors, for which we obtained the elite and viremic controllers. and especially Kathleen Lin and Vicky Roy from Elite Alter's lab, who performed the IgG biophysical experiments. But without the study participants, we could not have even conceived the study. And finally, I would like to thank the organizers and you for your attention today. And with that, if you have any questions, if I exceeded my time, feel free to contact us. And if you have any questions, I'll be very happy to take them now.
Thank you, Sankeet. That was an outstanding presentation, and you did not exceed your time. You were right on time, Sankeet, so very well done. Okay. We would be happy to take questions for Sankeet from the room or from the audience. Please use the Q&R function on your platform and ask questions. Let me maybe ask one first. Very interesting talk. I was just curious if you've ever characterized the accessory protein activity to measure, for example, how the nefs from these patients would function for ADCC in this cohort. Has that ever been done, Sankeet, for this cohort?
Yeah, that's a great question. So we are not, so we work from the immunology point of view, but I know that Dr. Andres Finzi's lab was pivotal with, I think it was Nirmin and Jeremy, if I'm not mistaken. They published a series of papers, 2016 or 15, 16 and 17 especially, where they isolated, and also Bruce Walker's lab. They did not use this specific cohort. However, what they also did was that they isolated NEF and VPU from elite controllers and compared it with those who were progressing, and they observed that elite controllers had a lower NEF activity for downregulating CD4 and KG2D ligands, BST2 for VPU especially, and they were pivotal in doing that. However, we did not study that from the point of view of the virus from elite controllers and viremic controllers, but they were the ones who did that, yes.
Okay, very interesting for potential future studies if you're interested. Any more questions from the room for Sankeet? Okay, I don't see anything. I'm just going to refresh my platform to make sure that I'm not missing anything. We're doing a quick refresh. Okay. Well, I will thank you for that excellent talk, Sankeet, and let's give him a nice applause. And at this point, I will pass the microphone over to my colleague, Dr. Nicole Bernard from the McGill University Health Center. Dr. Bernard.
Thank you. So the next presentation is going to be given by Julien Van Grenevege, and I'm not sure I'm pronouncing it correctly.
It's almost that.
Oh, good.
Not a big deal.
So I'm glad you're still here. And you're going to tell us about autophagy-dependent mitochondrial metabolism in the context of control by elite controllers towards successful cures.
Okay, do you hear me?
Yes, we do.
Okay, so I'm starting. First of all, I'm really happy today to present to you those new data about metabolic features, and I am going to take the opportunity of this 10-minute long presentation to remind you why it's so important to study the immunometabolism in the context of natural HIV-1 control, and especially among the groups of elite controllers. And I'm going also to take the opportunity to bring on the table a new notion that is provided in HIV context, is the metabolic plasticity. If you are interested about the full story about our data, you can go on PubMed, and there is 2 autophagies, and we published in 2021. Okay, so why the elite controllers are so important in the context of HIV cure? As it was saying this morning, despite the fact that ART for antiretroviral therapy is successful to bring a viral suppression in long term, it's absolutely not a cure. And also those individuals which are under treatments are not able to restore fully their memory and specifically their antiviral HIV T-cell responses. And when you take for granted that all the cures which are on the table today require a strong anti-HIV immunity, especially about CD8, and this in the context to avoid any re-explosion of the virus once the treatment is stopped, you can consider and it's easy to grasp that conceptually speaking, the heart-treated patients are probably not the optimal candidate for cures, or at least to ensure successful cures. And in this context, the elite controllers, which are individuals with natural, completely natural, without any treatment, control of HIV, could bring on the table new strategies to help those individuals and the other PLW. So what is the difference between elites and the rest of the groups of HIV-infected individuals? But despite the fact it's a really heterogeneous groups, and some of them, around 35%, have genetic advantages, all of them maintain a very strong anti-HIV immunity despite years of infection and persistent HIV-induced inflammation. And this is particularly about the polyfunctional cells and also the CTLA activity and the production of IL-21, which more and more the time goes, and more we found that it's a very important cytokines. And when you look on your right, the schematic show you that basically what's happened is that despite years of infections, elite HIV controllers are able to preserve a lot of mechanisms, a lot of molecular and metabolic features, despite, once again, years of infections. And the last and recent metabolomic and transcriptomic studies seems, or at least indicated and confirms, that elite controllers are the real metabolic program. And it was the topic of my research a few years ago. So what is the immunometabolism? The immunometabolism is basically the interplay between the immunity, the immune responses, and how the cells and the effector cells are going to use their cellular metabolism to provide a maximum of protection. And this in the context of different environments. Because remember, in the context of chronic HIV infection, and even in patients on the heart, there is still some residual and chronic inflammation. So why we were interested about autophagy? Autophagy is, as you can see here on the schematic, it's a catabolic process, which vulgarly it's in 3 phases. And you can see here that basically you are generating vacuoles which are associated with lysosomes. Those vacuoles are called autophagolysosomes as such, and you can block their activity by baphyllomycin A1 and as such blocking the autophagy in the latent phases. And the reason why we were really interested about autophagy is because autophagy, it's another mechanistic pathways associated with the modulation in the metabolisms, and especially in the context of tumors, when glucose becomes scarce in the environment, they use autophagy to basically have more lipid and protein contents to fuel the mitochondria. So believe me, I am not going to show you those data today, but believe me, what I'm going to say, we confirm that indeed the autophagy status by using different approaches were higher among elite controllers in both their memory anti-HIV CD4 and CD8 compared to the patients on their heart. And their immune functions as polyfunction, CTL, and IL-21 productions were autophagy and energy-dependent processes. In our hand, we use a 6 hours activation model where we had 2 kinds of activation. Either we activated the overall memory population with anti-CD3, anti-CD28, or we decided to use a pool of GAG peptides to stimulate the HIV-1 clones. But today, I am going to show you some metabolic assessments that we did. So due to the fact that we need a lot of cells to do those measurements, I am going to show you what was done with memory and memory CD8 and memory CD4 activated polyclonally. So we had the question that does autophagy provide additional resources? And in the context of elite controllers. And what we found out that there was a kind of dichotomy between memory CD4 and memory CD8. Memory CD8, we found out that they use preferentially the lipid content versus the memory CD4, which use the proteins and especially glutamine, glutamate to fuel the mitochondria. And when we were interested about this, first we looked at the memory CD8, and my previous student, Hamza Lucy, used the imaging flow cytometry and developed an assay to measure and assess the lipophagy activity. Basically, during this 6 hours activation status, we use either or not baphyllomycin to have accumulation of lipid content. And by using the imaging, so the ability of the device to show co-localization of signals, we use a dye to stain the lipids. and a dye to stain the autophagolysosome vacuoles. And as you can see here on the right, there is much more autophagolysosome vacuoles, including lipid content in elites and in HIV neg than in ART. And here you have the values and we got with the machine. We did the same experiments on memory CD4 and we didn't find any differences. And we find also a really basal low levels of using the lipid content in CD4. However, when we were interested about the proteins and we collaborated with McGill University, we use their GCMS platforms in order to detect and quantify the intracellular levels of both glutamine and glutamate in those individuals. And as you can see here, the elite controllers and HIV neg has a much more higher concentration, intracellular concentration of both glutamine and glutamate, in contrast to ART, who has a significant and drastic reduction. So as I was saying, it's a question of metabolic plasticity. The cells is using other resources than glucose to provide fuel to the mitochondria. And it's a feature already shown, as I was telling, in tumor cells. So now that we confirm that autophagy indeed in elite controllers was able to provide additional metabolic resources to fuel the mitochondria, we use the CAHR technology in order to see whether or not autophagy had a real impact on energy production and mitochondrial respiration. And as you can see here on the right, this is the kind of kinetics that give you the machine. Basically, what's happening is that you are measuring the oxygen consumption rates within the mitochondria, and during the cycle of measurement, the machine, the device, is adding different mitochondria modulators, including the oligomycin, known to be a potent inhibitors of ATP synthase. So as such, you are able to get two values: the SRC for spare respiratory capacity, which basically is the optimal ability of your mitochondria to breathe, and the ATP-linked respiration. So once again, I am not going to show you all the data, but what you can see here on the left are the data was generated with memory CD4 T cells activated 6 hours in elite controllers. And we added or not, in black it's no treatment, in white it's treatment, in cultures, different inhibitors. You have the baphyllomycin to block the autophagy, and you have Beptes and R162 to block the glutaminolysis, which is the seroconversion and the use of glutamine. And as you can see here, is that indeed all the energy productions involve both autophagy and glutaminolysis, and in both SRC and ATP-linked respiration measurements. I put in red also the information. We didn't find any differences when we use specific inhibitors of glutaminolysis within CD8. However, when we use the etomoxir, which is able to block the transportation of new lipid content within the mitochondria, we completely shut down the energy productions. So what has to be learned about elite controllers? Because it's nice, it's beautiful to say that the elite controllers are better than everyone, that they display strong immunity, they are able to preserve and conserve some anti-HIV responses. But the idea is to use this knowledge to be about to apply it in the other group of PLW, and especially in the context to reinforcing antiviral responses before ART cessation among PLW. And what we found out in the same studies that we were able to re-educate the system, not fully, not in the levels of elite controllers, but significantly by using either IL-21 with CD8 and CAHR, which is an inducer of AMP kinase, which is an inducer of autophagy. And our autophagy was AMP kinase dependent. So finally, I don't know what is the time. I don't know if I took too quick, but anyone. Finally, I want to acknowledge the fact that these studies, and I put in plural, will not be able to be published without the help of Zalusif, who was a perfect student to me and was proactive and developed, as I was saying, different platforms and protocols. Especially, it was the first one to use the imaging flow cytometry to assess the lipophagy activity among primary cells. And he was able also to develop protocols and to adapt the protocols of CAHRs to be able to work with primary cells, which, believe me, was a pain in the ass at first. But anyway, I want also to acknowledge and thank the AIDS and research networking and Dr. Jean-Pierre Routy, which helped me a lot during my career. Of course, the Institute Armand Frappier, with different platforms allowing us to do great science, I would say, and collaborators, especially the MIR platforms from McGill, who help us for the metabolomic features, and of course the financial supports that you can see here and help me to move forward my research, despite the fact that unfortunately I don't have the CAHR here. But anyway, thank you for your attention, and if you have any question, I am really happy to answer your call.
Thank you very much, Julien. You're perfectly on time. So are there any questions from the audience? And while we're waiting for questions, perhaps I could ask you something. Could you comment a little further on the differences between CD8 T cells and CD4 T cells in the elite controllers?
Yes. So basically, the situation of elite controllers were within their memory, CD4 and CD8 T cells, it's similar to what's going on with HIV-neg individuals. It's a kind of preservation of the use of autophagy and the metabolic features. But what we found out by using different kind of specific inhibitors, metabolic inhibitors, during our cultures, and thereafter by the ATP production measurement, is that we have a real dichotomy of the metabolic pathways. CD8 are using, I am not going to say exclusively, because they are able also to degrade proteins, but lipids seems to be the major and preferential metabolic resources. In memory CD4, it was much more black and white. Lipids was almost not used. But we have to remember that it's in our model. It's 6 hours in vitro stimulation. And we found out for a fact that depending the conditions of cultures, you are changing a lot the intracellular metabolism. But you have a real dichotomy between memory CD4 proteins, memory CD8 lipids.
If I could ask another question about that. So you said that you did your stimulations with anti-CD28, anti-CD3, and with GAG peptides.
Yes.
So is that one of the conditions that really changes the profiles that you're seeing?
Can you repeat the question?
Well, that you did your stimulations of CD8 positive T cells with GAG peptides and with anti-CD3, anti-CD28. So anti-CD3, anti-CD28 is a much more potent stimulus than peptides.
It's comparable.
Is it?
It's the same levels, absolutely comparable. Basically, what you found out with the levels of percentage of autophagy, percentage of use of the different metabolic resources, it's exactly the same when you trigger the TCR receptors among memory and when you stimulate specifically the HIV clones.
Okay, any other questions? I don't see anything in the chat.
There is one in the chat, Nicole.
Okay.
Do you want me to read it?
Oh, does HIV impair autophagy? Excuse me, it must have just come up.
Yes, it's true. HIV per se, the viruses is able to play with the autophagy. It makes sense because as I didn't say this information, but if you looked at the autophagy schematics, I put aside all the functions, the main function known about autophagy, and there was a xenophagy, the use of autophagy to degrade viruses and other pathogens. So of course, HIV hijacks the systems to his own agenda, and he stimulates the first initiation phase because there is stuff associated with NF-kappa B, and is blocking the final phase of degradation because either the viruses will be targeted. But remember that in our context, everything is done under either elite controllers with natural HIV control, either in patients on the heart where we add antiretroviral molecules within our cultures. And we confirmed by ELISA, P24 ELISA, that we didn't have neither in elites, neither in patients on the heart, any viruses production. So it's really in a situation of viremia.
Okay, thank you very much, Julien.
You're welcome.
We'll move on to the next presentation, which is going to be by Chris Dominique Nasaki Yoka. on circadian clock machinery regulating HIV transcription in CD4 positive T cells.
Can you see my?
Yes, we can. Thank you very much.
Okay. Good afternoon, everyone. I'm Chris Dominic Ngasakyuka from Petroleum Kotas Lab, PhD student. I'm pleased to present our result on the role of circadian clock machinery in the regulation of HIV replication in CD4 T cells. However, tremendous progress was made to uncover molecular mechanisms regulating the HIV replication cycle, thus leading to the identification of potent antiretroviral drug included in the current antiretroviral therapy. Residual HIV transcription occurs residual HIV transcription occurs during ART and represent a major cause for chronic inflammation. Block and lock strategies are currently investigated with the goal to inhibit HIV transcription, in part by targeting the transcriptional machinery of the cells. Previous studies in our lab demonstrated that TS17 polarized CD4 T cells carry integrated HIV DNA in ART-treated people living with HIV and identified a unique molecular signature associated with HIV permissiveness in TH17 cells versus TH1. Among this signature includes component and regulators of the circadian clock machinery, in particular BMOL1, also called RNLTTL, the master clock, the master circadian clock transcription factor. BMOL1 forms an heterodimer with the clock protein that bind on E-boxes present in the promoter of certain genes to create rhythmicity at molecular levels via feedback loops in the expression of genes encoding protein. Among them, we have the RORs and the REV-ERB. In turn, ROR and REV-ERB bind to the ROR response element present in BMOL1 promoter to regulate its expression. Recently, it has been shown that the variation in cell-associated spliced HIV RNA in infected individuals on ART has a circadian basis. The group of Sharon Lewin demonstrated a significant relationship between cell-associated HIV RNA and BMOL1 expression. The authors hypothesized that the heterodimer clock BMOL1 bind to E-box sequences in the HIV promoter and positively regulate HIV transcription. Effectively, the existence of E-boxes in the HIV LTR have been reported in the literature. In the context of TH17 cells, the same mechanisms are conserved. Indeed, ROR gamma T as a master positive regulator of TH17 effector functions, while REV-ERB compete with ROR gamma T to bind to the ROR response element and negatively regulate TH17 features. While multiple studies were performed in mice, studies in humans are missing. Given the role of REV-ERB as a transcriptional repressor for BMOL1 and and RORC2, that is the human homologue of mice rogamati. Given the role of REV-ERB, we decide to target ROV-ERB, and for that we decide to explore the role of REV-ERB in the regulation of HIV replication and viral reservoir reactivation in CD4 T cells. So ROR and REV-ERB are nuclear receptors that bind natural ligand in the cells. In a recent study, our lab demonstrated that RORC2 blockade inhibit HIV replication in CD4 T cells, thus identifying RORC2 as an HIV positive regulator. To reach our goal, we decide to use commercially available REV-ERB agonists, SR9011, and the antagonist SR8278. So by comparing with RORC2 and replacing the expression of various genes, including the HIV genome, we hypothesize that REV-ERB will decrease HIV replication. By using the REV-ERB agonist, we found a decrease of RORC2 and IL-17 mRNA expression, as well as IL-17A production as expected. Thus, we confirm that this agonist interferes with TS17 effector functions in human cells, similar to mouse cells. To test the effect of REV-ERB pharmacological activation in HIV replication, CD4 T cells of HIV unaffected donors were exposed to replication competent HIV and culture in presence or absence of the agonist SR9011. We found that this agonist decreased HIV replication as reflected here by reducing the percentage of HIV P24 positive cells quantified by FACS and decreased viral particles in cell culture supernatant quantified by ELISA. We further assessed the effect of this agonist on viral outgrowth. So a simplified viral outgrowth assay has been performed with CD4 T cells of R-treated people living with HIV stimulated by TCR. Results generated with cells from 4 different donors demonstrate an inhibition, as you can see, on viral outgrowth in presence of this agonist in dose-dependent manner. So we further tested the REV-ERB antagonist SR8278 in the same viral outgrowth assay. In contrast to our prediction, this antagonist decreased viral outgrowth in CD4 T cells, as you can see, of R-treated people living with HIV. So to better localize the step of the HIV replication cycle affected by the REV-ERB agonist antagonist, we performed a single round infection using a VSVG pseudotype HIV construct. Statistical analysis revealed a significant increase in early, late reverse transcription, as well as integrated HIV DNA in the presence of the antagonist SR8278, indicative of a disconnect between the effect of REV-ERB on viral outgrowth from reservoir cells versus HIV reverse transcription and/or integration. So using the same model of infection, we consistently observe an increase in the intracellular accumulation of GFP in the presence of the antagonists, and indicative of an increase of HIV protein translation. However, there is a decrease of HIV P24 release in the supernatant. So in conclusion, we demonstrated that REV-ERB regulates Th17 effector function in HIV replication, but further studies should identify molecular mechanisms by which REV-ERB directly or indirectly is involved in various steps of HIV replication, particularly in transcription. So I would like to thank all Peton's Ankuta's lab, and thank all the collaborators. Also thank the founders and all participants to this study. So thank you to you.
Thank you very much. Nice presentation. Are there any questions from the audience? Okay, let me ask a question since I don't see anything yet. Is the idea behind this research to lock and block?
Yeah.
Okay, all right.
Yes.
So I think I understand. Perhaps I could ask another question. Since this is targeting Th17 cells, I mean, Th17 cells also have functions in terms of, control of other pathogens such as fungi and bacteria. Is it known whether or not these antagonists that are going to be able to block HIV transcription may have off-target effects that may not be very beneficial?
Some target effect, some off-target effect have been reported with the agonist, but it's not measured, and there is no reported in the literature of any target effect that is that could be bad for the
okay,
for the cells.
All right, I think I don't see any questions, any more questions, so we'll move on to the next presentation. Okay, the next presentation will be given by Alessandro Modica on the effects of immunosuppressants on HIV reservoirs in kidney transplant recipients.
There we go. Sorry, can you see my slides?
Yes, they're fine. Everything's
Perfect.
fine.
Thank you. So hello, everyone. My name is Alessandro Modica. I'd like to thank the organizers for giving me the opportunity to present my project focusing on the impact of immunosuppressors on the HIV reservoir after kidney transplant. Doesn't want to work. Okay. So the HIV reservoir is made of cells that are infected by latent proviruses. So the latent reservoir can persist in the body through cellular survival and proliferation. That proliferation can be homeostatic or antigen-induced. The vast majority of HIV reservoir cells underwent proliferation in their lifetime. We estimate it to be over 80%, but we know that that number is underestimated. Because of the immune dysregulation, medication intake, and possibly infection of renal cells, damages can be caused to kidneys in people living with HIV, and that will require a transplant. And that transplant will come with a treatment to prevent organ rejection through immunosuppression. that treatment will start on the day of the transplant and then continue for a lifetime. It will inhibit immune cells activation and proliferation. And more specifically, it will inhibit two different signals involved in T cell activation. The first one is the binding between MHC2 binding the TCR, and then the third signal, IL-2 binding its receptor. It has been shown in previous studies that different types of immunosuppressors can impact HIV. So anti-calcineurin can inhibit HIV replication, and then another one, mycophenolic acid, has been shown to decrease viremia. So because cell proliferation of latently infected cells contribute to HIV persistence, and the fact that transplant recipients will receive immunosuppressors that inhibit that cell proliferation and activation, our hypothesis was that immunosuppressive treatment will reduce the HIV reservoir through the inhibition of cell proliferation and activation. So to show it, we're lucky enough to have access to a cohort of 7 participants who are living with HIV and have all received a kidney transplant. They're all under heart, infected for more than 15 years, and have all received the same combination of immunosuppressors. The viral load are undetectable. The average CD4 count is of 450. The average age of the participants is 56 years old, and their time on heart is around 12.5 years old. So we receive 1 or 2 samples before the transplant that we use as a baseline, and then we receive usually between 4 or 5 samples following the transplant at regular intervals. So our goal is to assess the impact of modulation of cell activation and proliferation by immunosuppressors on HIV reservoir in people living with HIV after renal transplant. And to show that, we measured the impact of immunosuppressors on the immunological markers, on HIV DNA and cell-associated RNA, as well as the phenotype of HIV-infected cells. So first, to see the impact of the immunosuppressors on the phenotype of the TCD4 cells, we came up with a panel to assess the specific changes on different CD4 T cell subsets, as well as the expression of activation and proliferation markers. And then we also wanted to measure the humoral response, so the levels of antibodies against HIV during the immunosuppressor treatment. So the next few graphs I will show are the frequency of cells expressing different markers. So each donor has its own color. And what we see is in the months following the transplant, we do see a small, not small, but more significant decrease in the expression of HLA-DR, which is an activation marker, and Ki67, which is a proliferation marker. We also see a small but significant increase in the frequency of cells that are CD45RA negative, so the memory subtypes. And more specifically, that increase was seen in the central memory subtype. And then finally, the biggest impact we saw on CD4 subsets was for the Tregs that we identified as CD25 positive, FOXP3 positive, where we see in the month following the transplant a profound and significant drop in the frequency of that subset. Then when we focus on the antibody response, what we see here are the fold changes of the levels of antibodies found that target HIV envelope. And we do see that in the month following the transplant, we see a decrease that is maintained in most participants. However, that response is not specific to HIV response, because we did see the same trend in the drop in levels of antibody, but against coronavirus. So then for the quantification of the HIV reservoir before and after the initiation of immunosuppressors, from the cells of the participant, we were able to extract DNA and RNA. And from those materials, we did qPCR to be able to quantify the reservoir again, and also assess the transcriptional activity. So this is the DNA, the HIV DNA data that I also present in full change, which is a little more easy to understand. But what we do see is a small and not significant decrease in the size of the reservoir, or at least in the the amount of copies of HIV DNA found in the month following the transplant. However, that decrease was not maintained over time. And when we look at the transcriptional activity, we do see the same trend as the DNA, with decreases that are not significant and that are found in most participants, but are not, again, maintained over time. So then for our third objective, what we wanted to do is assess the effect of the immunosuppressors on the functional reservoir, so on the size and the phenotype of the functional reservoir, using a technique called HIV flow. So it's a flow cytometry derived technique developed by our lab, where from the cells of the participant, we do a CD4 T cell purification, we do a PEMA ionomycin stimulation, and then as any flow cytometry technique, we would stain these cells for specific markers. But here what we do more specifically is we use 2 P24 specific antibodies to be able to target and identify precisely P24 expressing cells. So P24 expressing cells are considered the functional reservoir because a lot of the cells where we can find HIV DNA, that DNA doesn't necessarily will be intact. And so this allows us to determine and identify the functional reservoir. So what these measures have shown is a no clear impact of immunosuppressors on the frequency of P24 cells. Something to keep in mind, we didn't have, because these were the last experiment we were able to do on the cells of these donors, and the cells available were pretty limited, which explains why for some of the time points we couldn't detect any P24. And then we wanted to focus on the composition of the reservoir that we found. So in the P24 positive cells that we were able to measure, we wanted to see the distribution in the different subsets. So this is an example of one of the donors where we have time points a month before the transplant, a month following the transplant, and then 2 years following the transplant. And as you can see for this donor, the composition of the reservoir remains the same. And even though the data is not here, it's true for most participants where we were able to have, we were able to find P24 positive cells. Again, here we didn't, for some time points, we can measure P24 cells, which complicated the longitudinal analysis. So in conclusion, immunosuppressive therapy decreases the proliferation and activation of CD4 T cells, which is concomitant with a modest and transient decrease in HIV reservoir markers in most participants. Our results also showed a modest effect of immunosuppressors on humoral response against multiple viruses, so HIV and coronavirus. And finally, all these results seem to indicate that the current immunosuppressive drugs that were given to the participants are inefficient to profoundly and durably affect the HIV reservoir, and suggest that the size of the reservoir is tightly regulated by homeostatic forces that will be difficult to counteract. I'd just like to finish with some limitations to my project. First of all, the small number of participants. We were very lucky, but it's n equals 7 and their heterogeneity. Also, the measures of DNA and RNA tend to overestimate the size of the reservoir, again, because it will take into account defective proviruses. And finally, as I said previously, the numbers of cells were pretty limited for the HIV flow experiment, which again explains why for some time points we couldn't detect any P24 cells. I'd like to thank all the members of my lab, all the people who helped me with these results, the people who are responsible of the cohorts. Of course, I'd like to thank the participants, without whom all this research wouldn't be possible. And I'd like to thank all of you for your attention. If you have any questions, do not hesitate.
Thank you very much, Alessandro, for this very interesting presentation. Can I ask the audience if there are any questions? Can you speak a little bit more about your HIV flow technique? Does it only identify cells that are actively producing virus, or does it also evaluate cells with latent infection?
No, so in this specific instance, no, it's only cells that are producing P24, which help us quantify specifically the size of the functional reservoir.
When you did your experiments, it was more like a before and after effect. Did you compare the level of immune activation with people that did not undergo a transplant and just stayed on antiretroviral therapy? I mean, I'm just wondering if the drugs that were used to prevent rejection further decreased the level of immune activation.
So we didn't have any. The only participant we used were the people in this cohort who received the immunosuppressors for the transplant. So we didn't compare it to other participants who maybe didn't receive that treatment or received an organ transplant.
It might be interesting to do that. I mean, that data is available if you were to access the primary infection cohort, for example, and you looked at people that were treated, you know, over several years. You'd have longitudinal data. And there are cells available, so you might be able to look at immune activation markers and see whether or not there's an advantage in terms of lowering immune activation in the people that are taking these anti-rejection drugs.
No, it could definitely be a good idea.
And then, okay, I'll look again to see if there are other questions. They're not, so I'm going to ask another one if I can. Yeah, I'm just curious, when you showed the results from the HIV flow, you have one person that's, you know, has like 100 cells that are infected, but most of them like close to 1. I'm just wondering if your conclusion regarding the drugs not lowering the, what you're calling it, the functional reservoir, is because you're kind of starting at close to nothing and you can't really go down from there.
Yeah, so actually actually I can maybe go back if you want. The green participant is sort of an outlier. Usually the results we would expect, of course, if we had more cells, it would be more precise, but usually you'd expect the number of cells in the functional reservoir to be close to what we're seeing in trend in most participants. The green participant is definitely now sort of not common in that sense. I did other measures just to confirm that what we were seeing were actually P24 and not and maybe not a I don't know, a mismatch or an issue with the experiment. So we're on track to trying to see if we can sort of understand why he had so many P24 positive cells, which is not common. It happens sometimes. It has been seen in people with where they had not a big reservoir when we see in terms of DNA measurements, but they do have a lot of P24 positive cells. So we're trying to understand why but we were able to confirm that the values we got were actually P24 cells with a simpler flow cytometry panel, just to confirm that data.
Okay, thank you very much.
Thank you.
Project.
Thanks.
Okay, so we'll go on to the last presentation of the session, which will be given by Hope Lapointe, who, change of topic a little bit, we'll be looking at humoral immune responses to 1, 2, and 3 COVID-19 vaccine doses in people living with HIV, receiving suppressive antiretroviral therapy. So everything looks good.
There we go. Hi there. Sorry, can you hear me?
Yes, I can hear you.
Sorry, I was having trouble switching to the cam and mic. All right, let's give this. I don't know. Can you see my slides?
Not yet. No.
Not yet. No. Okay. Now I'm having troubles. Sorry about this. Here we go. All right. How about now?
Yes.
All right. Thank you. All right. So hi, everyone. My name is Dr. Hope Lapointe. I come from the BC Center for Excellence in HIV/AIDS here in Vancouver, British Columbia. And today I'll be presenting our data on humoral immune responses to second and third COVID-19 vaccine doses in people living with HIV. So some quick background. We know that people living with HIV may be at greater risk of severe COVID-19 outcomes when compared to people living without HIV. We also know that they can experience blunted immune responses to certain vaccines, including vaccines for hepatitis and flu. So because of this, COVID-19 immunization is important and recommended in people living with HIV, but we also know that the responses to COVID-19 vaccines still remain incompletely characterized at this time. So for those reasons, our study objectives are to assess the long-term humoral immune responses to the second COVID-19 vaccine dose, but also look at the humoral response 1 month after the third COVID-19 vaccine dose. And we will also be looking at any differences in humoral immune responses against the different COVID-19 variants, including the wild type SARS-CoV-2 virus and the more recent Omicron BA.2 variant. So very briefly, our study population is made up of 99 people living with HIV and 152 controls who are people living without HIV. Now, people living with HIV in our study had to be on active antiretroviral therapy with a suppressed viral load for at least 1 year in order to join the study. At the bottom, you can see our figure, our timeline for sample collections relative to each vaccine dose. There are up to 8 sample collections across the study. And in this presentation, I will only be focusing on the time points at 1, 3, and 6 months after the second dose, but also 1 month after the third vaccine dose. The table here shows characteristics of our study population across both groups. Very briefly, I'll point out some key features. Notably, our people living with HIV population had a healthy number of CD4 cells at study enrollment. They had a median of 715. We also noticed that there was a difference between sex at birth between our different populations. Notably, our people living with HIV group consisted of more men at 88% versus only 33% in the control group. At the bottom, you'll also notice some information regarding vaccination status. Most of our participants across both study groups received an mRNA vaccine from either Pfizer or Moderna as part of their 2 initial vaccinations for COVID-19. In the people living with HIV group, there were a subset of participants who received a 1 AstraZeneca dose followed by an mRNA, and then another population who received 2 AstraZeneca doses. And you can see each of those account for about 10% for that group. Importantly, in British Columbia, for third doses, only mRNA vaccines were administered, either Pfizer or Moderna, and no AstraZeneca vaccines were rolled out during that time for the third dose. In this presentation, I'll be focusing on 3 different measures of humoral immune response. One will be total binding antibodies. The second will be live virus neutralization against wild type SARS-CoV-2. And the last one will be ACE2 displacement, which is a surrogate marker for viral neutralization. And this will be measured against both wild type virus and Omicron BA.1. So firstly, looking at total serum binding antibodies, and this is against the spike receptor binding domain. The figure on the right you'll see shows total amount of binding antibodies against the spike RBD. You'll see that there are 3 different groups. There are people living with HIV, there are controls, and there is also a COVID group. The COVID group represents all people who were COVID convalescent, irregardless of HIV status. If it didn't matter if they had HIV or not, they were lumped into that group if they experienced COVID-19 at that specific time point. Now, you can see at the time points that after receiving a second COVID-19 vaccine dose, most of the groups achieve almost 4 logs of total antibody levels, but we do see subsequent waning of antibodies over time across all study groups. Notably, we do see a slightly lower antibody level in the people living with HIV group compared to the controls and COVID convalescents. Another thing worth mentioning is that although we do see waning of antibodies across all groups, there was no difference in the rate of antibody decline between the groups. They all decline at roughly the same rate. So these data seem to suggest by univariate analysis that people living with HIV actually produce lower amounts of antibodies after a second vaccine dose when you compare them to controls. However, when you do a multivariable analysis and correct for other factors such as sociodemographic, health and vaccine related things, we actually see that HIV is not correlated with a lower binding antibody level. Instead, we actually see that other factors play a more important role. For instance, we see that age, number of chronic comorbidities, and having received 2 CAHR or AstraZeneca doses, for those who received 2 AstraZeneca doses, they had lower responses compared to others. We also did notice that if you had an extended dosing interval between the first and second dose, or if you had prior COVID-19, that you had better responses, which is supported by other literature. Now, if we look at peak immunity a month after the 3rd dose, we actually see that the 3rd dose actually increased total antibody levels by about half a log across all groups. And based on this graph on the far right, you can see the data for the after the 3rd dose. We're seeing equivalent levels of antibodies across all groups, with the caveat that most of them are now achieving the upper limit of quantification. So we can't actually measure beyond this. And for that reason, this analysis is a little bit more limited and we can't do multivariable analysis. However, fortunately, we are seeing that everyone has produced more antibodies when compared to the 2nd dose. And this is regardless of HIV status. Moving on to live virus neutralization. And this is looking at the ability of plasma antibodies to inhibit infection of target cells. And this is looking at wild type virus. In this figure, essentially the y-axis, the higher you are on the y-axis, the better your viral neutralization is. And here we actually see that people living with HIV mount equivalent viral neutralization compared to controls. And there are no difference between those groups, although people who've experienced COVID have better neutralization. We do see that long-term at the 6-month time point after a 2nd dose, about 52% of COVID-naive participants had very, very low neutralization ability. And it was actually below the limit, the lower limit of quantification. And this suggests waning of neutralization ability over time and is supported by the decrease in vaccine effectiveness that we see over time. Importantly, on the far right, you see again data after the 3rd dose, and we're seeing again a boost in immunity after receiving a 3rd dose. When we perform a multivariable analysis, we actually see similar factors that play an important role. HIV status, again, did not correlate with neutralization ability. Instead, it was age, number of chronic comorbidities, and having received 2 AstraZeneca doses. Looking at our final data set on ACE2 displacement, or a surrogate marker of viral neutralization. This is looking at the ability of antibodies to block the ACE2 and spike interaction and prevent infection. And this is looking at data for wild type and Omicron BA2. This figure is only looking at data 1 month after the 2nd and 3rd vaccine doses. Again, higher value on the y-axis indicates better ACE2 displacement or better surrogate viral neutralization. And again, we're seeing that there is no difference, no statistical difference between the people living with HIV and control group. They mount equivalent responses in terms of ACE2 displacement. However, we do see a very striking difference between the variants. We can see that for wild type, people are mounting very good ACE2 displacement, nearing 100%. However, Omicron is mounting a much weaker response, and this is supported by the reports of immune evasion for Omicron BA1. And again, we see that the 3rd dose does provide that extra boost in immunity. So in conclusion, we see that people living with HIV mount equivalent humoral immune responses against SARS-CoV-2 when compared to controls. And this is after receiving the second and third COVID-19 vaccine doses and adjusting for socio-demographic factors, vaccine, and health variables. We didn't see any difference in immune waning between the two groups. And we also did not see that the lowest CD4 count or the nadir CD4 count on file. This was not correlated with a weaker response in our people living with HIV group. Instead, the only factors that we observed for weaker responses were age, number of chronic comorbidities, and again, 2 doses of AstraZeneca as part of their initial vaccination series. This data is highlighting that third doses provide an extra boost and don't simply maintain immunity in both populations, people with and without HIV. However, we do see decreased response to Omicron BA.1, although the third dose still provides some benefit, and that's applicable in the current era with multiple Omicron subvariants. So key messages: in short, people living with HIV with a healthy range of CD4 count and on active antiretroviral therapy that is suppressive mount very strong immune responses to the second and third COVID-19 vaccine doses. And we see that the third dose still provides some benefit despite having lower immune benefits than compared to previous variants. Just to highlight some key variants, and then I'm done, is these findings may not be applicable to all people living with HIV, namely those who are not on therapy or have low CD4 count or are not achieving suppressive therapy. We also don't know the thresholds for immune protection for any of these measures. We don't know what provides optimal protection, particularly against the new Omicron subvariants. And importantly, we are still gathering more data on cellular immunity and also long-term immunity after the third vaccine dose. So I want to thank very much my team for helping us to get all this done, and most importantly, the participants who've been with us for over a year and continue to put up with us and provide all these samples. We're very, very grateful for all your help and contributions. Thank you.
Oh, thank you very much for that good news story. I think that we've got a question here from Dr. Gilgrass. She's asking she's commenting that this is interesting data, since we have now seen differences in immune responses dependent on Moderna versus Pfizer. And if mixed, are you able to add this to your variables?
We did look at mixed vaccine regimens if it was a single AstraZeneca followed by an mRNA vaccine. If I recall, I would have to look at the results. I believe it came up as non-significant, but we would have to look at that again. One limitation I would point out is that for this study, because we didn't have a high enough sample count, we actually did essentially group the mRNA vaccines into exactly that, just mRNA vaccines, rather than Pfizer versus Moderna versus AstraZeneca. So that's one limitation. Although we have performed some more recent analyses, and we are finding that Moderna, which is supported by other literature, Moderna is slightly superior over Pfizer in providing those humoral immune responses.
Thank you. Just out of curiosity, I know that you're using commercial tests to look at anti-RBD antibodies, et cetera. But I'm just wondering if it's possible, if you use diluted plasma, if you could actually, expand on some of the results that are really kind of smushed up at the top of your y-axis.
Well, one limitation, which was what we saw after the third dose, was like you mentioned, a lot of people are achieving either the upper or the lower bounds of our assays, and this is becoming one factor. And I believe we are looking into performing diluted plasma experiments for the ACE2 and the live virus neutralization experiments. But as for the total antibody measurements, we use the Roche Alexis kit, and that's the one with, we are already diluting the sample and achieving the upper limit of quantification. because of that, we've actually implemented another antibody measurement test. This one will only measure IgG rather than total antibody, but this one has a much higher threshold or upper limit, and therefore we can actually extend it beyond the 25,000, which is the current limit for the assay that I reported in here. So we are trying to further expand these, given that so many people are achieving these lower and upper limits.
Thank you very much. So we've come to the end of the session. I'd like to congratulate everybody and show my appreciation for how well everyone did in terms of keeping on time. And I think you all deserve a round of applause. Thank you.